Welcome to this overview of Cyrus Bench for drug discovery. Cyrus Bench, powered by Rosetta, brings you the tools for protein design and small molecule discovery for therapeutics, diagnostics, and other industrial uses. Rosetta has been around since the late 1990s. Rosetta is responsible for most of the major firsts in protein design and structure prediction. Rosetta continues to push the envelope in the area of protein design. Going back to the late 1990s, Rosetta was responsible for the first accurate protein structure prediction without homologs, using just the sequence. This represents a giant leap forward in the protein fold problem. Since then, Rosetta has been a regular winner in the CASP competition, which is a biannual structure prediction contest. Prediction methods are assessed by how close predicted models are compared to the known crystal structures, which are not made public until the contest is over. Rosetta has become the leader in structure prediction and is especially accurate when using homologous structures as templates. In the early 2000s, Rosetta researchers began to branch out into the area of protein design, and most interestingly, Rosetta was used to create novel protein structures from scratch. The first design of a novel protein was done in the Baker lab by a researcher, Brian Coleman, who now runs his own Rosetta lab. The proteins they created were called Top 7. They are the first man-made protein fold, which was a huge turning point in protein design. Soon after, the first design of a protein-protein interface was created. This took the concept of protein design and used it to show that pragmatic goals can be achieved with Rosetta design. They were able to mutate surface residues to create novel binding. This had obvious implications and energized a lot of new research projects. In 2008, Rosetta was the first to design new function in an enzyme, and in 2010, Rosetta was used to design a protein that inhibited flu pathogen by designing a protein binding site. Then in 2014, Rosetta was used to design a protein to bind a specific small molecule. This area of research has broad and meaningful research potential. This allows enzymes to be designed for new substrates and furthers the field of small molecule discovery. The next major Rosetta first is the design of a pH-dependent binder to an antibody. This shows Rosetta's capacity to not only model proteins with a parameter like pH dependence, but antibody modeling. And finally, in 2018, there was a de novo design of a fluorescent beta barrel. This is a great example of how Rosetta can computationally design novel function. Today, there are many labs that have built tools in Rosetta for protein design, and the use of Rosetta design tools by other Rosetta labs is more commonplace. This has led to a variety of approaches to protein design with a wide range of goals. While some people focused on stability, others looked into methods to improve solubility of proteins they were working on in their labs. Some looked to alter protein affinity, specificity, or selectivity to a binding partner. These are all design goals that can be done in Cyrus Bench or through a Cyrus partnership. All this protein design expertise led to a set of rules on how to design proteins from scratch. This allowed researchers to create much more complex types of designs. Protein design rules and Rosetta were used to create self-assembling nanocages. These are heteromultimers that have multiple interface designs. These fascinating proteins were not only designed to associate, but to snap together into a large cage. Researchers working on these hope to use them for cargo delivery in therapeutic applications. For structure-based drug discovery, the key factor in success is always the accuracy of your input structure. It's always preferable to get a structure experimentally, ideally a high-resolution crystal structure. But great structural information, of course, can also be found with NMR, cryo-EM, and others. These experimental structures can be great input for structure-based drug discovery, but often Rosetta can improve the structure's fine details that may be wrong in the experimental model due to crystal packing, non-native conditions, or other artifacts. If you don't have a structure at all, we can often provide a great structure using homolog proteins as templates for modeling. Rosetta has been shown to consistently create homology modeling structures that have a 2 to 3 angstrom accuracy range. And with a high-quality protein structure, you can move forward with structure-based methods with the confidence that you have good input. Protein design is one area where your input quality will greatly affect the outcome. When designing for thermal stability, it is particularly important that the core amino acid interactions be more accurate. When attempting to design increased affinity, it is the interacting residues that should be accurately modeled. Binding sites need to be precise when modeling docking to a binding partner or to an epitope. A common structure-based method is the creation of a library of potential sequences that will be used for affinity maturation. Rosetta can narrow down a giant library of potential mutations into a manageable set of mutations more likely to be hits. And for protein therapeutics, quality structures are necessary for identification and design for the removal sequence liability, such as a design to remove immunogenicity and make a protein less prone to aggregation and more soluble. Rosetta is also excellent at creating great structures with the necessary accuracy for structure-based small molecule discovery. Rosetta is often used in conjunction with Schrödinger, Chemical Computing Group, Mo, BioVia, Accelerus, or other structure-based small molecule platforms. Many of our clients use us to model the proteins, then another tool for features such as small molecule docking. So they have the best of both worlds. Also, once you've established the docking mode of a molecule with its protein partner, that docked structure can be loaded back into Cyrus Bench for further structure modeling and design. These are all things we are doing with partners in pharma. Rosetta has consistently proven itself to be exceptional at structure prediction, especially when building models with the help of homologs. Rosetta has created high-accuracy models for a wide range of use cases. In this example from the International CASP competition, the correct structure is shown in black. On the left in orange, you can see that the closest homolog has a similar structure, but variation in the loop is significant. To the right of that, you can see that competitor predictions, shown in purple and blue, were able to predict with reasonable accuracy. But to the right of those in green, you can see that Rosetta gets the loop with an exceptional degree of accuracy. This is a model with the kind of accuracy necessary for many structure-based drug discovery methods, and this is the same method that we use in Cyrus Bench. On the right, you can see how much accuracy was measured over a large number of structure predictions. The blue bar is the Rosetta method that you can find in Cyrus Bench, and competitor predictions are the green bars. For this, we are measuring the hydrogen bond recapitulation, which is a way of measuring details in structure predictions. In addition to structure prediction, Cyrus Bench is an excellent tool for protein design. Here is an example of design using Rosetta that came out of the Baker Lab at the University of Washington. This is gliadinase, which is being explored as a potential treatment for celiac disease. In celiac disease, patients react to alpha-gliadin found in gluten. It is particularly resistant to proteolysis, so the protein triggers an immune response and causes inflammation. The Baker Lab used an endopeptidase, shown in the left in blue. They docked alpha-gliadin to the active site and used Rosetta to design affinity to this new substrate. They created a library of potential gliadinases to test in the lab. Amazingly, they found hits, and with further improvements with experimental and computational approaches, they created an effective gliadinase with high activity. It was developed out to PVP Biologics in a partnership with Takeda, and this is now in phase 1 clinical trials. This work is exactly the kind of designs that can be created in Cyrus Bench. Here is another example. This is a design for stabilization of cytosine deaminase. This is a component of TOCA511 retroviral gene therapy for treatment of recurrent high-grade glioma. It is in phase 3 clinical trials with Tocagen. Again, this work originally started in the Baker Lab. On the left is the wild-type protein with the beta sheet that makes up much of the core in green. Researchers in the Baker Lab computationally sampled millions of potential point mutations and combinations of mutations and created a small library of potential hits. The most successful set turned out to be the 3 mutations shown in the center structures that are in spheres. The mutations shifted the melting temperature by 10 degrees. This relatively simple application is broadly useful in biologics or industrial proteins and is available in Cyrus Bench. Our last example is deimmunization. Therapeutic proteins are often found to cause an immune response. There are 3 examples, again from the Baker Lab. On the left, this protein was tested in phase 3 clinical trials, but was found to be immunogenic. The Baker Lab redesigned the protein in order to maintain its structure and function, but altered the sequence in the epitope regions. The design was found to be successful ex vivo in humans. In the center, this protein was used in order to treat muscular dystrophy, but was also found to be immunogenic. Testing of the deimmunizing design has shown to be successful ex vivo in humans. The initial work for this research is shown on the right, where GFP, known to be immunogenic in mice, was designed to remove those immune liabilities, then tested again and shown not to elicit the immune response in mice. This epitope prediction tool is available in Cyrus Bench, and of course our design tools can be used to change the sequence at predicted epitopes while maintaining a stable structure, and then the new sequence can be run on the epitope prediction tool again. Thank you for listening to this overview of Cyrus Bench. If you are ready to start a trial of Bench, just click our link and you'll soon be designing proteins like a Rosetta professional. and feel free to contact us for more information.